going to answer a different kind of question about process of diffusion. And what do we have here is a diagram which has starch solution and iodine solution. As you can see, actually it is an experiment to demonstrate a certain process. And so our first question is, state the process that being investigated. That's obvious. That process is diffusion because we are talking about something that's spreading more widely. Like iodine solution is going to separate wider through the starch solution. And remember, we are observing iodine solution instead of water as a solvent. That's why we called it as a diffusion. And the second question is explain the results in experimental setup 1. Now we are looking at that one. At the moment, as a solvent, we have iodine solution. And iodine molecules have so small molecular sizes, and therefore it would diffuse through the tiny pores of the whisking tubing membrane, as you can see, into the starch solution, resulting to the blue-black coloration. So once that iodine solutions diffuse into the whisking tubing, it's going to end up with a coloration, blue-black coloration. That is due to the chemical reaction between iodine solution and the starch molecules. And it's going to be this color after chemical reaction between iodine solution and starch particles. And how about starch molecules? How about starch particles? Starch particles are too large. Starch particles are too large. They're not too large, and they would not pass through the tiny pores. They would not pass through the tiny pores of the whisking tubing, so they're going to stuck there inside that whisking tubing. And accordingly, it would not diffuse from the whisking tubing into the boiling tube or into the beaker. So thereby, there would be no observable color change on the outside because there's no chemical reaction. They wouldn't meet at all. And last question is, what result would be expected if the experiment was repeated using starch solution which had been boiled with dilute hydrochloric acid for 5 minutes? So actually there would be no observable color change on the inside, either inside or outside of the for the both setups. The reason for this is boiling starch with hydrochloric acid would hydrolyze the starch into the simple sugars, starch solutions, into the simple sugars, like breaking down the complex sugar into the simple sugars like maltose or glucose, etc. So, and these simple sugars would not diffuse through the whisking tubing. Actually, they would diffuse through the whisking tubing because they are simple sugar. Because they are simple sugar. but it will show no color change with iodine solution because it's not a starch molecules or starch solution anymore.